Dear students, welcome to the 8th session of Histology, Animal Behavior and Applied Zoology paper prescribed by the Mangalore University for 4th semester BSc Zoology. In this session, we shall consider the study of histological features of mammalian pancreas. The pancreas is a compound gland that discharges digestive enzymes into the alimentary canal or gut and secretes the hormones such as insulin and glucagon, which are vital in carbohydrate, specifically the glucose metabolism into the bloodstream. Pancreas is an elongated organ of about 12 to 15 centimeters long and composed of three different parts: the head of the pancreas, the body of the pancreas, and the tail of the pancreas. The pancreas is located posterior to the stomach with its head in the curve of the duodenum. You can see here above there will be the stomach, and below that the duodenum will be there. In the curvature of the duodenum, the head of the pancreas is situated. The body and tail of the pancreas extend laterally, and the tail of the pancreas generally touch the spleen. Pancreas is considered as a mixed gland or a dual gland, as it acts as both exocrine as well as endocrine gland. I hope you already know about exocrine and endocrine glands. Exocrine glands are the glands with a duct system, and their secretions are transported through the specialized ducts. In contrast to the exocrine glands, endocrine glands are commonly called as ductless glands. That means they do not possess any kind of ducts. They secrete their secretions directly to the bloodstream. As an exocrine gland, the pancreas secretes digestive enzymes into the pancreatic duct, which transports the pancreatic juice to the duodenum. As an endocrine gland, the pancreas secretes hormones such as insulin and glucagon into the bloodstream. Only about 1% of the total weight of the pancreas acts as an endocrine gland. And this portion, you can see here some isolated structures colored with the purple and green. These are called islets of Langerhans. Hence, the endocrine part of the pancreas is called islets of Langerhans. An adult pancreas contains around 2 lakh to 20 lakh islets of Langerhans dispersed in between the pancreatic acinus. Now let us look into the structure of exocrine pancreas. The exocrine pancreas is a serous compound tubuloacinar gland. That means the exocrine part of the pancreas is composed of serous acinar cells which are connected to the tubes, hence they are called tubuloacinar, and they are also called as compound tubuloacinar because not only one acinus is present, there are many acinas which are connected with one another through the acinar ducts. Now let us look into the structure of a single acinar cell. The single acinar cell, you can see here at the end of this picture, you can see a pyramidal shaped cell. This is the structure of single acinar cell. The pancreatic acinar cells are pyramidal serous cells arranged around the central lumen. You can see here there is a lumen at the center, and these are surrounded by many acinar cells. They possess a round basal nucleus. You can see here the nucleus is round and it is present at the base of the cell. An abundant rough endoplasmic reticulum. Since they are secretory cells, they need lot of rough endoplasmic reticulum, and their cytoplasm will have numerous endoplasmic reticulum. Along with rough endoplasmic reticulum, an extensive Golgi complex as well as numerous mitochondria are also present, and along with many free ribosomes. This is the structure of a single acinar cell. You can see here there is a basal, circular or round nucleus formed by many mitochondria, rough endoplasmic reticulum, and Golgi complex. There are many zymogen or secretory granules and are membrane bound, densely packed in the apical region of the pancreatic acinar cell. You can see here the apical region, tip of the acinar cells. There are many densely packed membrane bound zymogen granules are present, which contains enzymes and proenzymes packaged in the Golgi complex. The endoplasmic reticulum, rough endoplasmic reticulum secretes, synthesizes and secretes these enzymes and proenzymes which are packed by the Golgi complexes, and these are vesicle-like structures which are called zymogen granules, which are membrane bound and densely packed and are situated at the apical region of the acinar cells. Their basal plasma lemma has receptors for cholecystokinin and acetylcholine, both of which are hormones. The duct system of pancreas. The initial intra-acinar portion of the intercalated ducts is formed by centroacinar cells which are low cuboidal with a pale cytoplasm. From the initial portion, the intercalated ducts converge into small number of intralobular ducts, which in turn empty into large intralobular ducts that empty their secretion into main pancreatic duct. So all the acinar cells present here will secrete their secretion to the intercalated duct, that means the small ducts which are present, and these intercalated ducts converge, all these converge to form an intralobular duct. That means In between two lobules there will be ducts. They are called intralobular ducts. And intralobular ducts all combine together. These are intralobular ducts all combine together and empty their secretion to the main pancreatic duct here, which is represented in the green color. The main pancreatic duct fuses with the common bile duct. See, the bile duct comes from the liver and the pancreatic duct from the pancreas. Both of them merge together, fuse together, forming a structure called ampulla of Vater. You can see here this common structure where the bile duct and the pancreatic duct merge together, fuse together, are called ampulla of Vater, which delivers the secretions of the exocrine pancreas and the contents of the gallbladder into the duodenum at the major duodenal papilla. And the opening of the hepatopancreatic duct consists of a sphincter called sphincter of Oddi, which regulates the secretion of both bile and pancreatic duct. The function of exocrine pancreatic secretions are: there are many enzymes that are responsible for digestion are secreted by these exocrine pancreas. And along with that, many other proteins and fluids are secreted. Let us look into them one by one. The first secretion of the exocrine pancreas is enzyme poor alkaline fluid. This enzyme poor alkaline fluid is released in large quantities by intercollected duct cells, stimulated by secreting a hormone in conjunction with acetylcholine. It probably neutralizes the acidic chyme as it enters the duodenum. See, we know that chyme is the food that enters to the duodenum from the stomach, where it will be mixed with highly acidic hydrochloric acid. Hence, chyme will be highly acidic, which may cause certain problems in the duodenum if it prevails as such. Hence, the chyme must be neutralized and the enzyme poor alkaline fluid performs this function effectively, making the chyme harmless to the mucosal layer of the duodenum. Along with this, the exocrine pancreas also secretes many digestive enzymes. These digestive enzymes are synthesized and stored in the pancreatic acinar cells. Their release is stimulated by cholecystokinin, previously known as pancreozymin, and constimulated by acetylcholine released by postganglionic parasympathetic fibers. Digestive enzymes are secreted as enzymes or proenzymes that must be activated in the intestinal lumen. These enzymes may include pancreatic amylase, pancreatic lipases, ribonucleases, deoxyribonucleases, proenzymes such as trypsinogen, chymotrypsinogen, procarboxypeptidases, and proelastases. In order to protect themselves from digestive enzyme trypsin, these cells manufacture trypsin inhibitor so that trypsinogen cannot be converted to trypsin within the cytosol of these acinar cells. Let us move on to the endocrine part of the pancreas, which are called islets of Langerhanses. Islets of Langerhanses are richly vascularized spherical clusters of endocrine cell masses surrounded by a fine network of reticular fibers. They are scattered among the acini of the exocrine pancreas in an apparently random fashion. There is no exact, uh, fashion for its scattering in the pancreas. It will be present everywhere, and they are scattered everywhere in the pancreas in between the acinas. And these are richly vascularized cells. They are highly vascularized with lots of blood vessels in between, masses of cells surrounded by a fine network of reticular fibers. There will be clear separation of acinar cells and the cells of the Langerhanses. And these islets of Langerhanses are composed of several types of cells that can be differentiated from each other only by immunohistochemistry techniques or by the use of special stains. They produce several polypeptide hormones, but each cell produces only one type of hormone. So this is very important. Though the cells of these islets of Langerhanses look almost similar before staining, they can be distinguished only based on certain immunohistochemistry techniques, which are highly advanced techniques where the specific antibody is raised against the specific component secreted by these cells and are coupled with some chromogens so that we can distinguish the cells based on the kind of their secretion. One speciality of the islet cells are each cell produces only one type of hormone. They don't produce multiple hormones, and hence it will be very easy for anyone to go for immunohistochemistry and look into the type of the cell. There are three types of very important and conspicuous cells present in the islets of Langerhanses. These cells are named as alpha cells, beta cells, and delta cells. The alpha cells secrete glucagon, which acts to elevate the blood glucose level. Beta cells secrete a very important hormone called insulin that is responsible for decreasing the blood glucose level. The delta cells secrete a hormone called somatostatin, which inhibits the release of hormones by the nearby secretory cells and reduces the motility of the gastrointestinal tract and gallbladder by decreasing contraction of their smooth muscles. This is all about the histology and anatomy as well as the function of pancreas. For any further information, you can refer these textbooks. Thank you.